Okay, so good morning. Thank you to the organizer to invite me, and I will present to you today the Eyevensys technology. How does that go? Whoops. Okay, I'll get there. So Eyevensys is a small biotech company which is headquartered in France, in Paris, but also incorporated actually here in Texas. It was founded by Professor Francine Béard-Cohen, ophthalmology based in Paris. The technology is based on three components. First of all, it's taking plasmid DNA, they are encoded to produce therapeutic protein. This plasmid DNA is injected with an ocular device, which is designed here in the middle, as you can see, directly in the ciliary muscles, and then it's linked to a pulse generator. And by doing electrotransfection, that's what the pulse generator will do. This will allow the plasmid DNA to enter in the cells of the ciliary muscles, and the therapeutic proteins will be produced directly in the vitreous and in the choroid, which is very important. We have been in the clinic. We validated the technology in non-infection uveitis. Validation means just making sure we can do it, we can deliver, we can produce plasmid, and it's safe. The injection takes a few seconds. The administration takes a few seconds. Of course, we have demonstrated the technology in a bunch of animal models. What does that mean? We have been able to show that we can construct and code plasmid to produce any type of protein, enough to have in animal model, enough to have efficacy and safety. Also, what we have been able to demonstrate, we can construct plasmid and produce 2 therapeutic protein. And this is actually what we're going to do in our wet AMD program. What also we have shown in animal models is we have sustained delivery for at least 6 months, maybe 9 months based on the models. And this is just after 1 administration. The first indication that we were targeting was not infection uveitis. This is with an anti-TNF alpha. The goal here was really, it was a tactic to validate the technology. What we wanted to do is go in the clinic as quickly as possible. And we did in France, UK, and United States to demonstrate that we can do this technology and it was safe. We enrolled 18 patients in a dose escalating studies. And these were patients with late-stage non-infection uveitis, which were not responding to any treatment. What we did show in these 18 patients is that, A, we were able to administer the plasmid. We were able to show there was no real severe AE. The AE were mainly subconjunctival hemorrhage and very similar to an IVT injection. What we did show is that in the highest cohort, we had at least half of the patients who showed some either improvement in vision or reduction in edema based on OCT or reconstruction of the easy zone. And that's shown in 2 of the best cases here. Again, I'm not saying that we have demonstrated efficacy. This was not a study designed to show efficacy. So what did we learn from that study? A, that we can do electrotransfection. We can administer plasmid in the ciliary muscle. We can produce protein. It's safe. And of course, as you know, with devices, we had to collect also information so we can improve the design of a device. And that's the new generation here. So next step, what do we want to do? We want to go in wet AMD and geographic atrophy. For the wet AMD program, we want to take advantage of the possibility to combine 2 proteins under 1 plasmid. So 1 plasmid encoded to produce 2 proteins. Of course, we have aflibercept that we choose. But also we are producing decorin, which is a protein with anti-angiogenetic, anti-fibrotic, and anti-inflammatory characteristic. And the goal here, what we want to accomplish, is obviously reduce the number of retreatment after 1 injection, and also help prevent fibrosis in these patients. Right now, we are in the NHP studies, and the hope is to go in IND enabling phase very quickly. The second project, we're targeting GA first, with a possibility for RP. In this disease, we know that iron is very critical to protect for the health of the retina. But we also know in this disease, when the cells are dying, there's production of extra iron. So the idea here, and this has been extensively published, the idea here is really to build the plasmid to produce a native protein, which is called transferrin, which is an iron chelator. And what we want to do is, we have done several models, of course, but what we want to do here is target GA first, and if funding is allowing us to do also in parallel RP, whatever mutation it is, and basically what we want to accomplish is slow down progression of the disease with one injection repeated every 6 months. So in summary, what is the Eyevensys advantage of this technology? We can produce protein in a sustained manner for at least 6 to 12 months, maybe clinical trial will decide. It's safe. We can build plasmid to produce 2 proteins. We know we can do it. It's not a technique which is very invasive. It will reduce the burden on doctors and on patients. It's non-viral, so no risk of inflammation due to the virus. It's easier to manufacture. I'm not saying plasmid is easy to manufacture, but definitely much easier than viruses, and doesn't require subretinal surgery. The pipeline I just talked about. The team is very lean with a lot of experience in ophthalmology. I'm sure you will recognize some of us, and same for the board. Thank you very much.